Well, we're in the home stretch, so it is my pleasure to present the progress from the hepatotoxicity working group. I am presenting today in lieu of my co-chair, Dr. Steve. Um, sorry, I'm having
Ferguson.
a brain moment here from Steve Ferguson. Due to the government shutdown, obviously he was unable to present. So, really sorry for that. Our mission is the same as everyone else, so I'm not going to spend too much time on that. Here's a list of our current members. As I mentioned, Steve and I co chair this working group. And like all of the other working groups, we have membership from industry, academia, and government. And I won't, go over the list in too much detail. This we started with the original 13, and what you see highlighted in green here is, you know, reports in the literature, either in vitro or in vivo, in animals or human case reports of liver liability, liver toxicity, or potential botanical drug interaction. And so, some of the data is, maybe more solid than others, but nonetheless, we wanted to explore what we saw in the screening tools that we chose for looking at hepatotoxicity. And so we are really fortunate, I think, in the hepatotoxicity working group that we have two models involving hepatocytes, 3D hepatocytes and 2D hepatocytes. We're focusing just on human hepatocytes, but of course, hepatocytes are available from multiple species. But these models have been used for, in some cases, with the 2D for decades. And so we have tremendous amount of historical knowledge, both in terms of, chemical data, chemical testing data, some, probably more than maybe some of the other assays, complex mixtures, including botanicals, but also some of the tools, the screening tools that we're using. They've just been used forever, so we're fortunate for that. Um, and we are looking at on the right hand side. We're looking at a number of assays. Some of them we're looking consistently between 3D and 2D. And everything from just your basic cytotoxicity markers, ATP depletion, LDH release, other markers of cytotoxicity, which you would perform in any anything you're testing. You always start with cytotoxicity in order to delineate the appropriate dose response curve that you want to use. But we're also including more modern techniques like the transcriptomics and cell painting to look at effects on cell structure. So those are, you've heard some of the other working groups are using transcriptomics, but we're using it as well. And so that's a newer kind of screening tool that we're anxious to see how it works. So I'm going to start just presenting some cell viability data for the 2D hepatocytes. And this work was done by Dr. Albert Lee. And of course, certainly an expert in 2D human hepatocytes. So in the top pane, you'll see LDH release data for both, which I'm just showing you milk thistle and kava here. Milk thistle as our negative control, kava as a botanical where there have been some reports of potential hepatotoxicity. And then in the lower pane, we're looking at an MT glow assay, which is a way of measuring cell viability. And what you see across this broad range, very broad range of concentrations, for milk thistle, a little less so for kava, is that we do, although the data look a little messy. What we see is certainly a trend in increasing cytotoxicity with the higher dose levels, and that drop off that you see at the end, at the highest dose level, is pretty typical when you get to very high dose levels and you start to just have dead cells. You kill cells, and so that's not too uncommon. And you see this consistent between the LDH release in the top pane and the MT glow in the bottom pane. A little less toxicity, cytotoxicity for kava across the dose ranges that we tested. And, just to kind of proactively address the question about how we chose concentrations, similar to what others have suggested. At the onset, we wanted to test the broadest concentration range that we could, and then have data available to then go in and look more specifically at how these levels correspond to what's used in dietary supplements. And so I would kind of refer back, if you can recall, to, I think, Catherine's response to that question from the DART working group. It's a very similar approach that we will take. So next, we'll look at cell viability in 3D hepatocytes, and we see very similar profile. It's not really until you get at that higher dose level that you see the LDH release, and a similar profile. Milk thistle seems to be more sensitive at that. We're testing a much higher dose range than what we could test in kava. Oftentimes, we were just limited by solubility in terms of how high we tested. We tried to test to the limit of solubility. So next, we move on to looking at cytochrome P450 inhibition and induction. We'll start with the 2D hepatocytes. And we only looked at 3A4. And that's primarily because we really just, from a resource limiting standpoint, we chose to look at one P450 to begin with, 3A4 being the one that's probably most involved in, of course, drug metabolism, and has the probably highest levels in liver. We chose to look at 3A4. And so with induction, we did not see any induction across the dose range that we tested with milk thistle. So that is not unexpected. That was we thought would be an appropriate negative control. But we saw a really strong induction response with kava, a stronger response, in fact, than what you see with St. John's wort, the far right-hand pane. And St. John's wort, of course, is known to be a positive control for P450 induction, 3A4 induction. And so this was kind of a surprise to us. There's not a lot of hepatocyte induction data for botanicals. Actually, most people, just don't go that far. So we hadn't seen this before, and we're quite interested in these results. And now looking at inhibition, we again are showing milk thistle and kava here, and looking at 3A4 inhibition, and we really see no results. No inhibition. You see a decrease in the viability for milk thistle, but that's really due to the cytotoxicity that I showed in that early slide. So no inhibition from either milk thistle or kava, but definitely induction from kava. Now, moving on to the 3D primary human hepatocytes and looking at inhibition, we see a similar profile for inhibition. No inhibition from either milk thistle or kava. Uh, maybe a slight inhibition at the upper treatment level, but again, you're starting to maybe start to see a little cytotoxicity. Uh, we used ritonavir as a drug positive control, a known potent inhibitor. And so, you can see what really potent inhibition looks like. So we try to use botanical positive controls when we can, but we also use, drug controls because of just that broad and historical knowledge that we have in hepatocyte models. Now, Steve also looks at induction using a transcriptomics approach. And so he's looking at various genes uh, for things like PXR, CAR, and the regulatory mechanism of induction. And what you see here is an interesting way to just look at potency of induction and of based on gene expression. So, both milk thistle and kava cause clear changes in some of those genes that he was looking at. Milk thistle in blue there on the far right hand side obviously was the least potent of the botanicals that we looked at. That is as expected. And then kava was the most potent, as you can see in red. And you'll see data here from Yohimbe. I'm not speaking in depth on that, but we also see some interesting induction with Yohimbe, and so we're following up on that as well as a case study. So this provides some potential mechanism of action information as well. So and makes it for really easy way to kind of get an overall picture of potency and maybe help one to prioritize what you might want to follow up on in more definitive assays. And then lastly, I'd like to present just briefly some work. We also are interested in looking at potential reactive metabolite formation. And this is just a brief slide on work conducted by Dr. Richard Van Breemen's laboratory at Oregon State University. And he, incubated kava with human liver microsomes, either with or without human liver microsomes, and showed the formation of glutathione conjugates with both milk thistle and kava. And so you see the conjugate with the phytochemical constituent taxofolin, or some of the metabolites of kava lactones. And so, again, this may help address some of the mechanism of action of some of the liver toxicity that one might see once glutathione is overwhelmed and there's potential for reactive metabolites. So in discussion, we think that the liver enzyme leakage, the LDH release, worked well in 2D cells for botanicals. And this is, probably not too surprising. I mean, this, I've certainly used LDH release in a number of studies that I've conducted personally with various botanicals. Steve felt that the 3D models, are less sensitive to cytotoxicity with these botanicals. Kava-induced 3A4 at levels comparable or higher, actually, than St. John's wort. And so we really want to follow up with that to see if the results are clinically relevant. So, we have various ideas of some work that we, might like to do. And, if funding was available, we'd even like to do a clinical interaction study if we could, just because, this hasn't been shown. Omics approaches and reactive metabolite data have been generated, and we think that those approaches can aid in providing mechanism of action insights. And like everyone else mentioned, we're ready to now begin, really looking at how we can use ADNI data to, better position the data, understand in vitro to in vivo extrapolation, clinical relevance of some of our findings. And we're also excited, that work has been launched with some advanced liver models with a few of our botanicals. This is being done at FDA by Dr. Kirsten Ekstrom, who's a member of our group. And so we're excited to see the results from her work. And we, I think, as others have mentioned, we've started to look at some constituents. We'd love to follow up on further on some of the constituents of some of the botanicals, particularly where we're seeing some interesting results, and test those. And we have a number of manuscripts in preparation. This is a manuscript that we put out some time ago. And then we also had a session 2 years ago at the Society of Toxicology, really focused on botanical induced toxicity and liver injury and botanical drug interactions. And it was really well received. Had a really nice attendance and a lot of discussion from the audience. Obviously, a lot of interest in these endpoints, and so we published that as a workshop report. We presented some of these data at the recent SOT and the ICSB meeting, and we've also presented the data to the FDA in a presentation that Connie and I gave earlier this fall. And we are considering a number of next steps. Obviously, we like others, we're trying to determine what do we do with all of these data that we've generated. Too much, as Christine said, I think too much for a single publication. So we're looking at how we parse out the data into multiple publications. Do we, some of the data on just a single botanical are probably interesting enough for a publication. And like others, we want to collaborate with other groups, and also looking at future work. Understanding the clinical relevance, of course, is key, especially with botanical drug interactions. I think, we see a lot of reports of in vitro interaction potential, but it often doesn't translate to clinical relevance. And we want to look at potentially some additional cell models. We've talked about organ on a chip. And one of the things that's not listed here, but many of our members are interested in incorporating gut models into testing. And so we've actually, reached out to a few groups to understand their gut models and how we could incorporate, the gut and the liver, because I think it's really key to understand, the two together. And then, as with others, lot to lot comparisons of botanicals to really understand assay responses. So with that, I will stop sharing. And I think we're ahead of the game. And so,
Great. Thank you, Amy. We have one question in the Q&A already. So with regards to the transcriptomic data, have they been used to look at only at the single genes? Has any pathway level analysis been performed? Specifically considering there are AOPs for liver toxicity, has there been any attempt to look at whether the transcriptomic profile provide evidence for the presence of key events that may suggest downstream adverse effects?
yeah, no, we really haven't gotten to that depth yet. Clearly, that's something that we want to do. And in fact, we've really only looked at just a very brief look at the three botanicals I think that I presented. Um, that is one of the things that we were actually hoping to begin to deep dive on in our day two of what we had hoped to be day two of this annual meeting as a working group to really deep dive into the data there. But I think really dependent on Steve to help lead that, and obviously with the situation that's going on, we were unable to make that happen. But maybe whenever we get that day two rescheduled, we'll be able to focus in on that. But clearly, absolute great suggestion and where we need to go.
Great. Thank you, Amy. We have another question. Is it possible to determine which constituent of botanical plays the primary role in CYP inhibition or induction? Is it also valid to investigate whether there are interactions between different constituents?
Yeah, well, so I mean, certainly, I think it's possible, maybe quite labor intensive, but definitely, I think we will start to, in some cases where we're particularly, we have interesting results, say for the kava induction results, for example. Um, if we wanted to really try to understand what is the phytochemical constituent or constituents that's you know primarily responsible for that induction, we could certainly start to look at that. But you can imagine it becomes quite complex, right? Because do you just choose the do you just choose the phytochemical constituents at the highest concentration in the extract to look at, or who's to say that maybe it's a minor constituent that's the primarily involved in the finding that you're seeing. So that can be a little bit tricky, but that is something that we are interested in trying to do some work on. So any suggestions that anybody has, again, I mean, these are, I think, a great way for those of you, participating who are maybe not currently actively involved in the Botanical Safety Consortium. There's always ways to get involved with some of these ideas.
Great. Thank you. Are there any additional questions? Feel free to raise your hand or put them in the chat.
I guess I would, just to, kind of build on that as I'm thinking, I think the next step, right, is to really start to incorporate that ADME and understand the relevance of the concentration range of where we see those findings to determine, are we just, so much higher than what is really realistically used? Would we even bother to go down that path? So I think that's the next thing we need to think about is just, how do we determine whether or not to pursue some of these additional studies. So I think that's probably what needs to happen next is just the relevance piece.
Great.
May I
Thanks,
had
Amy.
a thought and want to get your opinion on, for hepatotoxicity, there's so many cell types, right? And there's so many cell
Yeah.
setups. We probably can't do everything, but could you maybe speak at a high level on like Hep G2 versus Hep RG versus the work we did in
Yeah.
primary human hepatocyte?
Yeah. Well, I think we really like the human hepats, the primary cells, right? We like the primary cells because we have this historical body of knowledge. They're used. They're the gold standard. Certainly in the pharma industry, and so we really like to use those cells. So I would kind of hate to um, utilize our time and resources towards other, you know, cell models that aren't primary unless it was to help maybe in work with other working groups who are using some of those cells. If there was that collaboration that could be made. But you reminded me of, something else I wanted to mention, and that's another thing that we're considering, and we've talked to folks who have you're right, the liver is very heterogeneous. I mean, we're kind of biased towards hepatocytes, but there's many other cells that are there, particularly Kupffer cells, the resident macrophages, the endothelial cells. So that's another thing that we've really considered, is looking at co-cultures of primary hepatocytes with other cells, especially so much of liver toxicity may be immune mediated. So I think that becomes very important.